First, I'd like to thank the organizers for giving me an opportunity to present our work. Today, I'll talk about this new computational method that we've been developing to find synthetic lethal partners from large-scale primary tumor data. So, quick primer on synthetic lethality. It refers to a genetic interaction, typically between a pair of genes, where knocking down each gene individually has no effect on cell viability. But if you knock down both genes in the pair, then the cell dies. And this is an attractive paradigm for treatment in cancer, because if you look at the figure over here, if one of the genes in the pair is a cancer mutation that has lost some important functionality, now if you can inhibit its synthetic lethal partner, then you can selectively kill only the cancer cells while keeping the normal cells alive. And probably the poster child of clinical translation using synthetic lethality is the case of BRCA1 mutated breast cancer and the use of PARP inhibitors. So ever since this interaction was discovered, there's been a lot of efforts to try and find more of these synthetic lethal interactions. And typically people use experimental screens to try and find these methods. So you start with a pair of cell lines, an isogenic pair where one has a mutation and one doesn't, and then you use these lentiviral shRNA libraries usually to systematically knock down a set of genes, and you look for the phenotype that you're interested in. In this case, prefer more death in the presence of the mutation versus absence. So while this sounds conceptually very simple and desirable, there are several limitations. Some of them are technology limitations, like you could have off-target issues for the shRNAs. So you're not sure whether the phenotype you're observing is because of the particular gene that is supposed to be knocked down by the shRNA. And there are also other issues like limited coverage of genes. So most of these standard platforms don't actually have shRNAs to knock down each and every gene. And then there's the issue of the fact that you're using cell lines for many of these studies. And so cell lines may not often reflect in vivo tumor evolution. And a lot of issues have come with the fact that the results that you have shRNA screens done with one cell line, and when you move to another cell line, you cannot recapture the same results. And there's also other issues, such as like they're restricted by the availability of cell lines, especially with some of the newer mutations that are coming up. And a parallel development that has been happening is the fact that there's been an explosion of primary tumor data, as all of you in this room probably know, because of projects such as the TCGA. So this figure is from, I think, last year's TCGA progress report. And so on the x-axis, you have the various tumors, and on the y-axis, you have the number of cases shipped. And if you sum up all the cases, there are about 11,000 primary tumors, as opposed to about a maximum of 1,000 cell lines. And interestingly, many of the newer mutations that have been discovered, such as epigenetic mutations like IDH1 and 2 or DNMT3A, and then the spliceosome mutations are not very well represented in cell lines, whereas you have a lot of primary tumor cases with those mutations. And another finding, which is sort of expected, but it has been reinforced, is that many of these mutations are shared across tumor types. So there's an advantage to looking across many types of tumor and try to infer some of these relationships. So that's kind of the backdrop where we got started. Our high-level hypothesis was that we would primarily rely on pan-cancer primary tumor data to identify these synthetic lethal partners. And the specific hypothesis was that synthetic lethal partners of a mutation X will be present in more copies in tumor samples which have X. So one manifestation of this is the following, which is the synthetic lethal partner of a mutation will never be deleted in tumor samples with X. So if you look at a scatter plot between, say, mutation X and deletion of a gene A, which is supposedly a true synthetic lethal partner of A, and in the scatter plot, each dot here is a tumor sample. So what you would expect to find is the upper right quadrant would not have any samples, because tumor cells which both have X mutated and A deleted would ultimately die and hence be underrepresented in the tumor population. So, and for us to find these relationships, we use an algorithm called Boolean implications, which is a very simple but highly scalable and effective data mining approach. And this is what we call a high-low Boolean implication, which states that if X is mutated, then A is not deleted. Now, another manifestation of the same, of the hypothesis could be that the synthetic lethal partner is selectively amplified in tumor samples with X. So if you go back to a scatter plot here, what you would find is that B is only amplified in the presence of the mutation, suggesting a form of dependence. And again, this is a very easy implication for us to pick out. This is what we call a low-low Boolean implication, where if X is not mutated, then B is not amplified. And obviously, both of these relationships that I showed you, there could be many reasons why they happen when you analyze mutation and copy number data. So we put in additional filters using gene expression to at least remove some of the obvious false positives. So this is what the pipeline looks like. We have called the tool MiSL. And so we start with mutation and cancer type, and the type of cancer, which could be, say, acute myeloid leukemia. And then you try to pick out tumor types where the mutation frequency is above a certain threshold. At this point, we are using the PanCan11 data that has been put out by the TCGA. And then in a pan-cancer setup, we look for these implications, these high-low implications and low-low implications. And of course, we also do a filtering with gene expression, because many of these copy number alterations are often not functional, so they don't result in any changes in expression, and we want to restrict only the functional ones. And so after this analysis has been done in a pan-cancer setting, now we come back to the specific cancer types, say acute myeloid leukemia, and we would like the gene to be overexpressed, the putative synthetic lethal partner to be overexpressed in the mutated samples versus the wild type samples. So we start out with with all of the genes, and then whatever survives all of these all of these steps is what we call the synthetic lethal candidates, or the MiSL candidates. So when I first implemented the pipeline, my first obvious thing was to make sure that we are getting a reasonable number of candidates for all of the recurrent mutations in the various tumors. So if you have too little, that's not good, and if you have too many, that's again not informative. And so when we were getting somewhere between 10 and 100 candidates, depending on the frequency of the mutation. So then the question was, are we actually picking up anything real? And in this case, it's a little hard because there aren't that many experimentally confirmed synthetic lethals. So what we decided to do was to compare with existing shRNA data, even though we know it's noisy, but the thing is, if we get some concordance with them, with shRNA data, it suggests that we're actually in the ballpark of picking up real things. So my first comparator was to look at shRNA data for the IDH1 mutated AML, and this was generated by one of our collaborators. So they had set up the system over here where they could express either the mutant IDH1 or the wild type IDH1, and then they transduced it with an shRNA library that had about 5,000 genes. And then after treatment with docs for about 12 days, they looked for shRNAs that were underrepresented in the mutant population versus the wild type population. And the idea being that the genes corresponding to the shRNAs that were underrepresented in the mutant population are the synthetic lethal candidates for the IDH1 mutation. And so using the system, they were able to find what, for all practical purposes, we think is a true synthetic lethal interaction between the IDH1 mutation and this BCL2 family members. And they have gone and actually validated it extensively in vivo and in vitro, so I feel confident saying that's a true synthetic lethal interaction. So what happened when we compared their data to ours? So first I looked at just comparing the synthetic lethals from the shRNA screen versus the MiSL candidates. So this is what we get from the shRNA screen, and these are the MiSL candidates. And first of all, we see a statistically significant overlap, which is reassuring. And then interestingly, we actually pick up BCLW over here. And so this is what it looks like in the MiSL pipeline. So BCLW deletions are mutually exclusive with the mutation. The deletion itself is functional, so deletion results in lowered expression. And then finally, in AML, you find that in the presence of the mutation, BCLW is actually overexpressed. So that was kind of, step one of validation, and it was reassuring to get that with data that our collaborators had. Next, we moved to data from Achilles, where they have a lot of shRNA data. And so this is a summary plot of comparing Achilles data with our results. So what I did was I ranked the genes according to essentiality in KRAS mutated colorectal cancer, And what we found as per a GSCA analysis is that the KRAS mutated MiSL candidates came up towards the top of the list. And this happened to be, and this was a statistically significant result over here. And this happened to be true for two other major KRAS, two other major colorectal mutations. So at this point, we are feeling, given two different types of sources of shRNA data, we were able to find concordance between our computational method and the experimental screens. So we were at a point where we thought we were actually finding some real things. So now we sort of switched gears and started looking for more novel interactions. And so we came back to IDH1 mutated AML because that's what our collaborators were interested in. So this is the list of MiSL candidates. It's about 80 candidates. And then we looked only at we looked for drugs that inhibited some of these targets, basically, and from a variety of sources. And we came up with a list of around 20 drugs. And then we did MTS assays where we treated both the mutant and the wild type for about 48 hours with the drug, and you calculate IC50s. So this picture here is a summary plot of what we saw. The x-axis is the drugs, and the y-axis is the percentage ratio of the IC50s for the mutant versus the wild type. And so if you have a value of 100, it means there's no difference between the mutant and the wild type, so no synthetic lethality. And ideally, you want to be towards the lower left corner over here. So again, reassuringly, we find ABT199, which actually inhibits the BCL2 family members, suggesting that this assay is actually working. And what popped up as an interesting novel drug is this drug TOFA, which is a very specific inhibitor of one of the MiSL candidates that we had. So we are following that up with more experimental work over here. And so TOFA inhibits this gene ACACA, which is acetyl-CoA carboxylase. And so both with drug treatment as well as with shRNA knockdown of ACACA, we find that the IDH mutants have a very strong proliferative defect compared to the wild type. And we see the results also in primary AML cells, where the mutant cells require much lower doses of the drug compared to the wild type cells. So at this point, we feel somewhat confident that we have actually identified a true synthetic lethal interaction, and we are following that up with more in vivo studies to make sure that, to see if this could actually be a therapeutic candidate. And just a quick note on mechanism, just to make sure that this was actually plausible. So both IDH1 and ACACA are part of our major metabolic genes, and they're part of the TCA cycle. And so according to published literature, the IDH, in the presence of the mutant, the conversion of glutamine to palmitate, which is fatty acid, is severely impacted. And fatty acid is needed by any proliferating cell, especially something like a cancer cell. And so one of our hypotheses was that fatty acid amounts would be lower in IDH mutants. And so we looked at the amounts of the precursors, malonyl-CoA and acetyl-CoA. And sure enough, we find that for both of these metabolites, in the mutants, the amount is much less than in the wild type. So our working hypothesis is that the IDH mutant cells are struggling to make fatty acid, and now when you go and you knock down ACACA, it serves really as a death knell for this particular type of cells. So just to bring it back to MiSL, so MiSL actually did not have any information about pathways. It was able to infer these relationships just by looking at mutation copy number and gene expression patterns. And again, we expect MiSL to be a pipeline that works for other mutations and other cancers. So this is an example in kidney cancer where we find a novel synthetic lethal interaction between VHL mutation and glutamine. And again, in this case, interestingly, glutamine is actually amplified selectively in the mutated cells. And this is our validation over here, which I can go over in detail with anybody who's interested later. And this is the case in breast cancer, again, where we have where MiSL discovers a synthetic lethal interaction between the PIK3CA mutation and AKT1, and we are able to validate it using a variety of assays. So just to summarize, so I introduced a novel method to mine synthetic lethal interactions, and we are solely using primary tumor data. And we think this is beneficial for two reasons. First, it leverages the growing set of primary tumor data sets. We expect more and more of this data to be available as it gets used potentially in clinical pipelines. And also, we are picking up these interactions which are based on in vivo selection pressure, so hopefully they would be more more likely to be translated in the clinics. And so far, our validation experiment suggests that we are able to both recapitulate known interactions and also find novel interactions. And there are plenty of other extensions to this work, such as drug repurposing, finding double knockdowns, etc. So that brings me to my last slide. Thanks to my PI, David Dill, and also a lot of our collaborators in the different diseases who actually who stepped in to do the experiments and funding sources. Thank you.
In the work you had where you were comparing to the Achilles data with the colorectal
KRAS,
yeah.
Right.
Did you have to restrict your training to the colorectal samples, or is it a pan-can KRAS that then you looked up in Achilles?
So I do pan-can, and then I actually kind of bring it back to colorectal, where I look for higher expression in colorectal specifically. So there is a specific cancer-specific filter at the end.
And then one last question. Your method, you talked about the low and the high-low.
Right.
Isn't it possible that you could both be amplified or not deleted? And can your method pick that up, or do you just break those into two cases?
If it's significant in both cases, it would get picked up. Okay. Like, so if the deletion is, if there's a significant mutual exclusion with deletion as well as a significant amplification, like a subset relationship with amplification, we would pick that up for sure. Yeah.
Thanks. Great talk.